Welcome to this presentation on analytical techniques used in peptide sameness testing. Peptide therapeutics are increasingly used in modern medicine, but ensuring that each peptide drug is consistent, pure, and structurally identical to the intended product is extremely important. Even small molecular differences can affect the safety and effectiveness of the drug. In this video, we will explore the key analytical techniques scientists use to confirm peptide identity, structure, and purity. These techniques help pharmaceutical companies maintain high-quality standards and meet regulatory requirements during drug development and manufacturing. Peptides are short chains of amino acids that typically contain between 2 and 50 amino acid residues. These molecules play an important role in therapeutic drug development because they can target biological pathways with high specificity. However, peptides are also highly sensitive molecules. Their structure can easily change due to environmental conditions such as temperature, oxidation, or chemical reactions like demidation. Even minor structural changes can significantly impact the safety, stability, and therapeutic activity of the drug. Because of this sensitivity, careful analytical testing is required to confirm that the peptide maintains its correct structure and composition. Sameness testing is a critical step in peptide drug development and quality control. It ensures that the peptide product being manufactured is identical to the reference standard or the originally approved drug. Regulatory agencies require this type of testing for the approval of generic and biosimilar peptide drugs. It helps demonstrate that the new product has the same identity and performance as the original drug. Sameness testing also supports quality assurance by ensuring that each production batch is consistent. In addition, it plays an important role in stability testing, where scientists monitor how the peptide changes during storage over time. To confirm peptide sameness, scientists rely on a combination of analytical techniques rather than a single method. Each technique provides unique information about the peptide's identity, purity, and structure. Mass spectrometry is commonly used to determine the exact molecular weight of the peptide and to detect possible modifications. High-performance liquid chromatography, or HPLC, helps assess purity and evaluate chromatographic behavior compared to a reference standard. Another important method is peptide mapping. This technique involves enzymatic digestion of the peptide followed by fragment analysis, allowing researchers to confirm the amino acid sequence at a detailed level. Mass spectrometry is often considered the first line of analysis when confirming peptide identity. This technique measures the mass-to-charge ratio of ions, allowing scientists to determine the exact molecular weight of the peptide. Because peptides can undergo chemical modifications, mass spectrometry is extremely valuable for detecting changes such as oxidation, missing residues, or other structural alterations. A commonly used approach is LC-MS, which combines liquid chromatography with mass spectrometry. This combination allows the peptide to first be separated and then precisely analyzed, providing highly accurate identification and structural confirmation. High-performance liquid chromatography, or HPLC, is one of the most widely used techniques in peptide analysis. It separates molecules based on their physicochemical properties, such as hydrophobicity or polarity. One important parameter in HPLC is the retention time, which indicates how long a compound takes to travel through the chromatography column. By comparing the retention time of a test sample with a reference standard, scientists can confirm similarity between the two. HPLC also helps detect impurities, degradation products, or synthesis byproducts, ensuring that the peptide sample maintains a high level of purity. Peptide mapping is a powerful technique used to confirm the sequence and structural integrity of peptide drugs. The process begins with enzymatic digestion, where specific enzymes cleave the peptide into smaller fragments. These fragments are then separated and analyzed using chromatographic or mass spectrometric techniques. Each fragment corresponds to a specific portion of the original peptide sequence. By comparing the resulting fragment pattern, often called a molecular fingerprint, with a known reference, scientists can confirm that the sequence and structural arrangement of the peptide are correct. Amino acid analysis is another essential method used to confirm peptide composition. In this technique, the peptide is first hydrolyzed, meaning it is broken down into its individual amino acids. Once the peptide is fully hydrolyzed, the amount of each amino acid is carefully measured. This allows scientists to determine whether the peptide contains the correct number and types of amino acid building blocks. This technique is particularly useful for verifying sequence composition and detecting missing or extra amino acids that may result from synthesis errors or degradation. In some cases, advanced structural techniques are required to fully understand the three-dimensional structure of peptide molecules. One such technique is nuclear magnetic resonance, or NMR spectroscopy. NMR provides detailed information about the atomic-level structure of molecules. It can reveal how atoms are arranged within the peptide and how the molecule folds in three-dimensional space. This information is valuable for studying molecular dynamics, confirming structural stability, and ensuring that the peptide adopts the correct conformation necessary for biological activity. In conclusion, ensuring peptide sameness requires a comprehensive analytical approach. No single method can provide all the information needed to fully confirm peptide identity and structural integrity. Instead, scientists rely on a combination of complementary techniques, including mass spectrometry, HPLC, peptide mapping, amino acid analysis, and advanced structural tools like NMR. Together, these techniques provide a detailed understanding of peptide composition, structure, and purity. By using this multi-technique strategy, pharmaceutical scientists can ensure that peptide drugs remain consistent, safe, and effective for patients.